Okay, this week in lab, you're going to do 3 exercises. They're all related to mitosis, meiosis, and also a karyotype, which that one's actually really cool. Not that the others are not. But so what you want to do first, of course, is download your answer sheet. I know that says Monday due date. Look, everything is going on in our lives. Do the best you can with the due dates. I'll work with you no matter what. But what you're going to do is you have some pictures here. These are onion cells. Those are very common. Our anatomy courses, we have 2 slides for mitosis. We have what's called a whitefish blastula, which are fish eggs in different stages of mitosis. I actually like those better. You can tell a little better with what they are. I gave you some examples of those. And then you have the onion root mitosis tips. Like right there, I'm seeing one already. My mouse is... there's an anaphase. The ones where you can see a nucleolus, like that one. Okay, here's the border. So there's the cell. There's square. And then you see the nucleus and the little dot, the dark, dense dot inside is the nucleolus. Like that's one that's an interphase, which is not mitosis. This one, you can see a cleavage furrow. That's actually a telophase. You really can't get this one wrong. You need to go through, do a good effort, fill out your chart, put where you think the predictions will be. How many cells do you think, and this is percentage-wise, add them up to 100%, but how many you actually think are going to be in each stage? Then you're going to go through and count them. So if you count 20 cells that are in interphase, you divide that by 65 times 100 will give you the percentage. So yeah, 20 divided by 65. That's how you figure out percentages. And then answer these questions. What stage of the cell cycle are the most of the cell tips in? How accurate were your predictions? Once again, you really can't really get those wrong. What's the function of mitosis in an organism such as a human? It's to replace damaged or dead cells. You lose skin cells every day. They have to be replaced. Meiosis. Now, I'm going to hit the control click. See how long this takes. It's going to take you to a website. Now, oddly enough, in the bonus lesson for this section, I had this exact video. So I would like you to watch this video. I go through the same thing with you.
Meiosis is the process that results in the formation of sperm.
And the video actually does give you the answers to most of these questions. Why is it necessary to reduce the number of chromosome number in gametes? Gametes are sex cells, eggs and sperm. I talked about this as well. You cannot have a 46 chromosome sperm penetrating a 46 chromosome egg. You'd have a different species, so the number has to be cut in half. That's one of the functions of meiosis. And as far as the other cells go, the other cells divide by mitosis. You can't have genetic gamete cells with 46 chromosomes. And I'm doing the word 46 for human beings. Every animal varies, but you'll end up with double the number of chromosomes in the zygote. Describe at least 2 differences between meiosis 1 and meiosis 2. I did cover that as well. I gave you these diagrams. This is actually a great one. This is meiosis prophase 1. You see how these chromosomes are bound together. It's called crossing over when there's some exchanging of genetic information. That does not occur during mitosis. You can see the chromosomes are not paired up. And then during metaphase, these are homologous pairs. See, they line up side by side, whereas metaphase of mitosis, they're on top of each other. Basically, like they're standing, like shoulder to feet, like somebody trying to climb over a wall or something. Once again, these are more like discussion questions, and then answer these. How many chromosomes are in each one of these cells? Same as before. It says bolded, underline the correct answer. Same there. And then here's our last experiment. I'll take you through the very first one. I wish this was a little bigger. It's a pretty cool website, but what you're going to do, this has to do with karyotyping. Any kind of chromosomal anomalies or abnormalities or deletions, they call it, or what's called non-disjunction. When chromosomes divide, it's disjunction. Non-disjunction is when they don't divide correctly. You end up with, say, like 321s in one cell. That's Down syndrome. So what you're going to do, go down here to start, click on, and it tells you, before I do this, it tells you exactly what to do. So read over the material, click on the complete, well, first of all, you're going to click on patient histories, and then you're going to finish out these karyotypes. And karyotypes are where the chromosomes are removed during metaphase, they are stained, they're photographed, and then they're paired up with their homologous chromosome. You get 23 chromosomes from your mom and 23 from your dad. I made a comment earlier about you get one gene from each parent. You do for all of your genetics, you get a gene from each parent, and then there's some variability in there. So what you're going to do is click on the patient histories and see there's there's 3 here. Now, one of these is tough, and I'm going to do the first one with you, but patient A is a nearly full-term fetus of a 40-year-old female. Now that automatically, older females are more can be prone to Down syndrome because of the non-disjunction, the non-splitting of the chromosomes. And this is an amniocentesis that was done on this person. So what you're going to do, so here's the chromosome you're looking to pair up. You see how these are all paired, and you want to go around and find the one you think this one looks like. Goodness, I did this a year ago. So see that one? I'm pretty sure. So look, I'm going to put my mouse here. See how it turns into a hand? And if it's not the right one, like over here, watch. It will tell you. See? So you got to try again. That guy looks like this one. There we go. This one looks like that one. This one, okay, it looks like a number 5, so I'm going to click on that. It says, now that you've paired up all the chromosomes, where does this extra chromosome belong on the karyotype? Okay, and they've actually underlined with blue the numbers. Let me see here. Let me click on that again. There it is. So I did good. The writing is very small. It says, congratulations, you successfully completed the patient A's karyotype. Now, it asks you to write in the matches, like what was chromosome A matched with number 5, chromosome B matched with number 12. Now, scientific notation on karyotyping is you would write, like mine, I don't have any extra chromosomes that I know of. Human beings have 46. I'm 46XY. This person has an extra chromosome, and they're female. It said that in the initial thing, amniocentesis. So her karyotype notation would be 47XX, and then sometimes you can do plus 21 or an extra 21. Like it says here, for example, 47XY plus 18 indicates the patient has 47 chromosomes, is a male, and has an extra autosomal chromosome. Autosomes are the non-sex chromosomes. So make sure you write that correctly. So you would put the number of chromosomes, you designate male, female, and then you put like a plus. In that person's case, it's a plus 21. What diagnosis? When does meiosis? Okay, when during meiosis does the occur? Sorry, bad English here. Does this occur that results in this karyotype? Be specific. Yeah, it definitely be when they're dividing up, when they've been paired up and they're splitting. They're not let's just say that an extra 21 means one cell got 121, and then the other cell got 3 21s. Okay, go through and do B. So what you would do is go down here. When there's the excuse me, your completed one, click on it again. Here's patient B. Ooh, looks like that one, and then go through and do all of those, and then fill out the rest of your lab form. All right, and that's your lab this week.